prokaryotic DNA fork. Let's jump into it. Here we have an example of what's of prokaryotic DNA, which can be seen in such prokaryotic organisms such as E. coli. It is a circular DNA, unlike eukaryotes, which are linear, and undergoes what's known as theta replication, in which one origin of replication, also known as ORIC, has two replication forks moving in opposite directions to create the replication bubble. Let's zoom in on the replication bubble and see what's happening. Bubble has zoomed in and focused. As you can see, there are two replication forks in opposite directions of one another. There's also a leading strand and lagging strands. The leading strand is 5' to 3', and on the parent side, and the lagging strand is 3' to 5'. Due to its anti-parallel nature, on either side they will be inverse of one another. Let's go onto this replication fork and start talking about all the enzymes at place. During initiation, there are 3 enzymes needed: helicase, gyrase, and single binding proteins that are the most important. Helicase is like a zipper. What it does is it breaks the hydrogen bonds on the double helix, giving off 2 single strands of DNA. This can cause some problems though, such as supercoiling, which is over-rotation of the strand at the head. Gyrase prevents this by binding slightly ahead of the replication fork. Another issue is these 2 single strands are very unstable and want to connect back to one another. So single binding proteins, as seen here, bind to the single strands and prevent this from happening, stabilizing them. On the leading strand, it synthesizes through continuous replication. So a single RNA primer is needed at the base, as DNA polymerase cannot bind to a free OH. After this RNA primer is placed, DNA polymerase 3 reads 3' to 5' and replicates 5' to 3', replicates continuously throughout the DNA cell, as seen here. That's called continuous replication. On the lagging strand, however, it uses what's known as discontinuous replication. So it starts off with DNA primase placing a singular RNA primer that is typically 10 to 20 nucleotides in length, and which leaves a small blank area that it's able to read for DNA polymerase, as shown here. DNA polymerase then fills this segment. This is known as an Okazaki fragment. As synthesis continues, DNA polymerase 1, also known as the editing polymerase, replaces these RNA primers with its proper DNA nucleotide. It fills up all the DNA bases except for a small sugar phosphate nick that ligase has to seal using a phosphodiester bond. DNA replication like this creates a semi-conservative model that is used in meiosis and mitosis and is the origin of all life.